These workshops started 2 years ago as part of a 5-day conference that the RSC CICAG organised on open chemical sciences. The meeting actually included open data and also open publishing. But the workshop side of things proved to be incredibly popular, and so we decided that we would continue to run the workshops every month using a variety or highlighting a variety of open source tools for chemists. We've now done 17 of these workshops, and they've been really popular. So we decided that we'd try and do a few more in 2022. So the first of these is on the tools from the protein database in Europe, and we're also delighted that Liverpool Biochem have continued to sponsor these events, and you'll be hearing a little bit about them in the interval. Just a couple of housekeeping notes. If you have any questions for the presenters, everybody is in listen-only mode, so please put your questions into the Q&A box, and we will then relay them to the presenter to answer them live. There will be a short break after 1 hour in which you give you time to have a walk around and stretch your legs. The recording of this workshop will be made available in a short period of time on the RSC CICAG YouTube channel. So without further ado, I'd like to hand over to David and Preeta to give us this workshop. Over to you. I think David's starting, aren't you?
Yes, thank you very much. So I'll just share my screens so that you have a presentation available. So hopefully everyone can see my title screen now. Just to introduce briefly ourselves before I kind of go on to the next session. So I'm David. I'm from the PDBe team, the Protein Data Bank in Europe team. My role is as outreach and training lead. That's partly responsibilities for outreach, but also I work in the curation team at PDBe as well. So dealing directly with the deposition of data that goes into PDBe and that side of things as well. So Preeti, did you want to just introduce yourself?
Hi everyone, I'm Preeti. I'm one of the bioinformaticians from the team, and I work on various data pipelines which expose data on PDBe and Pdbkb pages. And today I'll be talking to you about aggregated view of proteins and a part of our chem project later.
Yeah, thank you. So yeah, what I'm going to be talking about first is introducing you to the PDBe KnowledgeBase, which is a new resource that we've created. And specifically then going on to talk about the protein pages that we have for aggregating that data together. Before I get on with that, just give you a rundown of what this kind of schedule is going to look like, so you've got an idea of what's happening. So the exact timings might change a little bit, but this is the general overview. So I'll give a talk first on Pdbkb and the protein pages. We have some tutorials that we're going to make available for you as well. But I think during these sessions, it's going to be difficult to do that interactively. So what I'm going to do is introduce you to those tutorials, where to find them, and a bit about how they work. After that, we'll have a short break. After the break, then Preeti will talk to you more about this project called Biochem Graph, which is very ligand and chemistry focused. And what we're going to do is have a very short introduction to some designs that we've actually got for new pages, but we'd love to get some feedback on. So just to present those and show you how you can provide us with feedback. And then to finish it all, rather than having too many Q&A sessions in between, we'll have one final Q&A session at the end. Obviously, we can answer questions here or there as we go along, but it might be easier to address those questions in that final session, and then both Preeti and I can address those. Okay, so just to give you the overview of what the PDBe is before we get into Pdbkb. So the key thing here is Protein Data Bank is an archive of experimentally determined 3D structures. So these are always solved by a set of experimental techniques, and it means we have experimental data available to validate the models that go into the PDB archive. The other slight misnomer in the name Protein Data Bank is that it's not just proteins. Obviously, there's a lot of things that are bound to proteins, but we also accept polymers of nucleic acids, DNA, and RNA. One thing we've seen is the massive increase in data in the PDB. So actually, I think my stats are slightly off. I think we're over 190,000 now, getting towards 200,000 structures in the PDB. And what it means is this is a really big wealth of data that needs to be handled slightly differently. And that's one of the things we're trying to do with Pdbkb is change the way that we handle this data. Because one of the things about all of these structures in the PDB is each one of these PDB entries is an experimental structure based on one specific experiment. So if it's a crystallography entry, it's one crystal that was then put into a synchrotron, diffraction images collected, and then it was resolved. But quite often there'll be a lot of examples of the same protein that have been solved in different experiments, very commonly with different compounds bound. Perhaps this might be a particular drug target. So for example, on the right here, Aurora kinase, it's a target for trying to understand which drug fragments could bind, and maybe we could develop new drugs if we understand more about the binding of all these different molecules. So in this case, there's actually, for one specific protein, there's over 150 structures in the PDB, and these are bound to 150 different small molecule compounds as well. So one of the things we want to try and do is, rather than make people have to go to each one of these individual structures to get all that information, is try and bring it all together in one place. Now, the data that goes into the PDB archive is actually managed by a consortium called the Worldwide Protein Data Bank, of which PDBe ourselves are one of the resources. And we all work together on the deposition of data, and we manage a single piece of software called OneDep for deposition of data to the PDB. And once that's all done, the data is then released into specific PDB archive, which is essentially an archive of files. It's quite straightforward. What we then do, though, as different sites, is we then make that data available in different ways. I've also included the EMDB and EMRB data archives there, because they're also involved in the WWPDB project. But yeah, once we have these archives with that standardized data based on one worldwide project, we can then make that data available through our own separate websites and add additional value data to that, and use different tools to present that information to the user and hopefully improve the understanding of that information. So one of the things we add, both in the deposition process, but also even more after release of the data, is additional metadata. And this didn't quite work, this graphic, but it was supposed to show that there were every atom is defined in a coordinate file and positions of where it is, but actually all it shows is where these points are in a coordinate file. Until we actually get the context of how these atoms are linked, what information about small molecules and interactions between drugs and ligands and proteins, until we get that information, we don't have a full context of what this structure is actually trying to tell us. So one of the key goals at PDBe is to try and take this data from the PDB and add additional meaning to it with additional biological metadata or chemistry data as well. So when we've got all of this information, both in the PDB and then the additional information we add from PDBe and Pdbkb, what can we then do with all of it? Well, this is just a bit of an example of one particular entry, and actually what you'd see if you just looked at where all the atoms are in the coordinate file, which is essentially the kind of bare bones of what PDBe entry is. And you'd see something like this, and this is, as you see in the bottom right, this PDB entry 7LXY. But we want to add more context to this. For example, we might want to add structural context. In this case, we can define that this actually forms a homotrimer of this particular protein, and we can also see what other molecules might be interacting with it in this particular experiment. But we also want to add biological context. So this could be the name and the organism where this comes from. So in this case, this is a spike glycoprotein from SARS-CoV-2. And we can get information about things like macromolecular interaction interfaces. So where do these proteins interact together? Where are all the ligand binding sites that we can find from PDB data? In the cases where perhaps there aren't ligands found in PDB, are there any other predicted data that we can draw in to understand other ligand binding sites? And then, for example, information on variants. We can understand where the variants are across the protein and how these might affect all those different interactions. So we bring all of this information together to hopefully give a much more well-rounded context of what this structure is telling us and what it tells us about the function of the particular protein. So this is what we are trying to do with Pdbkb, is add this context through this community resource. And what we do is gather information from lots of other resources, because there's loads of resources which do very specialist things. They take PDB data, they analyze it, and they give all this additional information that's really beneficial for understanding the function. And so under the Pdbkb umbrella, we try and standardize this information and make it easier to access it all, and avoid people having to go to all these separate individual places to get this information, and kind of bring it under one umbrella. So how this looks like in terms of enriching the data in the PDB, well, on the left here we have PDB entries, which are deposited to the one-dep system in www.pdb. But we have these specialist databases that provide all these different functional annotations, and they could be dispersed amongst different PDB entries. They don't necessarily all map to all the entries which are relevant. So by then interconnecting all of these together with all the related PDB entries and highlighting where these different functions are across the whole protein, we can then show the context and enrich it with all this additional information from all these other functional databases. So that's essentially the process that we're trying to do with Pdbkb. And just to kind of highlight the scope of what we're aiming to do with Pdbkb and where we're going is to highlight it here. So the right-hand side here is kind of what we're looking to do with Pdbkb. At the moment, we have PDB entry pages already, which is the sort of typical way of looking at protein structures. We now have a new thing called aggregated views of proteins, which is what I'm going to present to you here. And we're also working on these aggregated views of ligands. So can we display the context of this work, context of what's in the PDB from the focus of the ligands themselves? And that's what Preeti will talk to you more about. I'll work on that in the next presentation. So our first iteration, I guess, of Pdbkb pages are the aggregated views of proteins. So as I mentioned, traditionally, most people have accessed PDB data by accessing a specific structure or by a bulk download of all structures with particular PDB IDs. Now, this is, if you know what you're doing, this is a good way to do it, but it's very time intensive. It needs a lot of specialist understanding, and it's likely to miss a lot of useful information that's available in other structures. So what we do with the Pdbkb aggregated views of proteins is we gather all the data up for a particular protein. Now, in this case, we base it on a uniprot accession because it gives us an easy way to link this data together. So for a specific protein, for a specific organism, we can gather all the information in the PDB for that example. So for example, if you look on the right here for this protein example, we give a 3D visualization at the top so you can see what the protein looks like. We also then show you that there are 30 different structures in the PDB of this particular protein. There are 7 different small molecule ligands that are found bound to this protein. And there are also 8 different interaction partners with this protein, other macromolecules that interact. And we've also got sections for additional annotations, for extension to find similar proteins that are very closely related, and also information about related publications as well. So this data is kind of presented through some interactive visualization techniques. So we try and display all the available data in a sequence related view to summarize it in a way that means you can get huge amounts of information, but summarized in one kind of easy to digest area. But because obviously this is protein structures, we also provide 3D visualization as well. And one of the newest things for this is a superposition process that we've added, which I'll mention in a bit more detail shortly. Okay, so firstly, just to point out the ligand section of these pages. So we have various different sections, but I'm just going to focus on a couple of them for this particular presentation. The ligand section of these pages defines all the small molecule compounds that are found bound to this particular protein in the PDB. And it gives you a list of the images of these molecules, how many entries they're found in. But then also further down, we have this, it's called a prop vista sequence viewer. And we can actually present all this data in terms of the protein sequence and where these interactions with these particular ligands occur within the protein sequence. Now, this is a really nice way to visualize it when you have huge amounts of ligands, because what we can do is clearly see where the common interaction sites are, and also see, probably more interestingly, where binding of slightly different ligands differs to the protein as well, and see what those differences are and maybe then work out why that is. So this is a really nice way to get an overview of all the available data for these particular proteins. You can also, if you're interested in a particular ligand, click on the images to get a 3D visualization of the ligand bound to a PDB entry as well. So the newer process that we've implemented is a superposition process. So for a given protein in a Pdbkb page, we use programs to superimpose the coordinates of these proteins onto each other and cluster them so we can find where similar protein structures, similar protein conformations occur. The benefit of this, as well as seeing these different conformations of proteins, is means we can also overlay where the ligand binding sites are for this particular protein as well. So let me just show you how you access those. So we now have buttons to access this on the summary page. So there's buttons underneath structures and ligands. So the first one of those, this 3D view of superposed structures, that shows your conformations and shows the different conformations clustered using the superimposition process. The second button, the 3D view of superposed ligands, that displays all the bound ligands amongst all the different clusters for that particular segment of the protein. So the issue in some cases is that we don't have data for the full length protein, in which case we might have to split into segments. So here's an example.
Can I ask a quick question about the superimposition?
Yes, sure.
Sure. Do you do that on the whole protein or say the active site?
So at the moment we're doing it on the whole protein. because this is based on the protein aggregated views, I say the full protein, we actually do it on segments, because in some cases for very long proteins, we actually have data for different sections of the protein, but not the whole one. But it doesn't focus necessarily on the active site, it focuses itself on the whole protein. That's one of the things that we're discussing towards the aggregated views of ligands, which Preeti will come on to, but there's also the option to superimpose based on the ligands that bind to the protein and see how the protein interactions differ as well. So yeah, at the moment we've kept it kind of full protein length, but we are looking at other ways we can superimpose that data.
Thanks.
No problem. So this is an example you might see, and this is one particularly busy protein example. There's hundreds of proteins in this example and lots of ligands bound to it. And you can see in pink all the different ligands found bound to this particular protein. And clearly from that you can see a cluster of where the active site would be because of this ball of ligands kind of bound to the center. So this, you might want to filter, if you're looking at very specific interactions, you might want to filter out ligands and maybe reduce the amount that are shown by default. The other interesting thing you can actually see here is common carbohydrate binding sites as well. So around kind of the outside there, you can see quite a lot of saccharide rings there where there's a common residue bound to a particular carbohydrate as well. So these give you these bits of information on superposition, which are quite nice to display where these common binding sites are. The problem at the moment is we sort of don't have a way of filtering this out yet by, I guess, ligands that you would consider important for different reasons. And that's something we're looking to do at some point. So we will have all the compounds, for example, which are reagents in the experiment that happen to co-crystallize with the structure, which might not be the interesting ones for research purposes, but it does give you a really nice overview of the data that's available. So the benefit of these pages is that hopefully they can answer scientific questions using such structural data. So rather than just showing people structures, we're hoping it gives them the tools to answer scientific questions they come with without having to, have that real in-depth understanding of protein structure. One of the reasons for trying to do this is that we know that the majority of PDB data users aren't structural biologists. So we want to make it as easy as possible for them to get useful functional data from these protein structures without having to integrate this data across vast amounts of structures and really understand the ins and outs of what a protein structure is. So these are things like, understanding which structures of protein form different conformations and how these differ, which small molecules interact with the protein, which macromolecules interact, and where those common binding sites and interfaces are. But also related to variants and diseases and things like that, how we can actually highlight where those variants are and interact, where they actually coincide with interaction interfaces as well, highlighting where they could cause problems with how the protein functions. So just before I move on, I just wanted to run through a particular case study, really, that gives you an idea of how this process kind of works and what kind of information you can get just by using Pdbkb pages. So this example is an adenylate kinase from E. coli, and this is found in 17 different PDB entries with 9 different ligands found bound to this entry. So from information about the ligand, we know that it catalyzes the transfer of a phosphate from an ATP to an AMP, and then that gives 2 molecules of ADP. So what information we can find on the Pdbkb pages? Well, if we go to the ligand section, we can find all of those 9 different ligands that are found bound. And I've shown a few examples here, and you can see that visually they fall into kind of 3 different forms, I guess, 3 different kind of general scaffolds. One is AMP, or monophosphate adenosine molecules. The other example I've drawn out here is AMP, which is essentially an ATP analog, but a non-reactive one, which is usually used in these crystallography experiments to allow us to capture those reactants before they react. And then the final one is actually a kind of conjoined molecule, which has 4 phosphates with 2 different adenosine moieties on each side. And so we can see those clustered by the visuals, but also if we look at the interactions, we can see where the interactions are similar between these different molecules. So in the case of AMP, we get a particular kind of interaction fingerprint, we could define it as, which is very different to that interaction fingerprint that you get for ANP, so the ATP analog. And then interestingly, you can see that the AP5 molecule, the one which is kind of a conjoined molecule of both of these separate molecules, you can see that the interaction profile for this particular molecule covers both those examples. So without even looking at the structure, we can see that AMP and ANP have different binding regions. However, AP5 covers both these regions. So we can kind of tell where these fit in the structure. And then on the right-hand side, we can show the superimposition of this particular structure, which highlights where those ligands are in the binding site and displays that, backing up our understanding of the interactions, that these form these 2 binding sites next to each other, which AP5 then comes across and conjoins. The other interesting thing we get from the superimposition is we can actually see 2 distinct clusters. And we only find this orange and closed cluster in cases where ligands, either the AP5 ligand is bound or that both these binding sites are occupied. So it also gives us an understanding of, we know that we need both ligands to be bound in order to create this closed conformation of the protein to lead to the reaction to happen. So this is all kind of information that we can get by looking at these PDBe pages, analyzing the interactions and looking at the superposition. So this is quite a rich amount of data before you even go into looking at publications and things like that. So before I finish, sorry, Chris, did you have a?
Yeah, there's a question in the Q&A. Is it possible to superimpose the structures of different proteins, such as protein isoforms?
So at the moment, on our pages, we're only doing it for single isoforms of the UniProt entry. What we are doing though is we're making the matrices required to create the superimpositions available. So actually, if you view any superposition data, you can actually get a link to the matrices to regenerate these superimpositions. And then obviously you can then take these and add on your own using your own kind of superimposition matrices. So yeah, it's not something we're doing yet across different proteins, but it's possible to do this by downloading the data from the PDB and then adding your own to it.
Okay, great.
So the final thing I want to talk about is something called 3D beacons, which is a new resource that we're working on to kind of expand the knowledge of what we know about protein structures beyond just experimental structures in the PDB. I won't spend too long on this, but just to highlight the reason for needing this. I mean, if we compare UniProt and the PDB, it's about a 1,000-fold difference in the number of proteins in UniProt compared to the number of protein structures that we have in the PDB. So such a tiny number of these structures actually have experimentally determined structures. What we are also seeing is that the vast improvement of predicted protein structures. So by taking those sequences and computing them to generate 3D structures, we can actually predict the structures for a lot of these proteins. And many of you may be aware of some advances in AI with protein structures, particularly for AlphaFold, which we are part of the AlphaFold protein structure database as well, which provides theoretical protein structures to fill in the gaps where we don't have these for known PDB entries. So what 3D Beacons does is it takes all of this structural information from these different resources and kind of links it together by a UniProt ID. So this includes data from PDB, the PDB archive, but also information from small angle scattering databases, AlphaFold DB, as I mentioned, Swiss model. We just added model archive as well, which is another large source of protein structure models as well. So it means that all of these different structural models are available for people to view and to improve the availability of structures of some kind for a particular protein. So 3D Beacons is an API that does work behind the scenes, and we actually integrate a lot of these into our Pdbkb aggregated views to enhance the number of models that are available there. But it is also available at 3dbeacons.org as well. And this shows you for a particular protein where all the different structural data is available. And it works very similarly to Pdbkb as well, where it shows you for which region of the protein structure there is data available and the different types of predictions that are available. Okay, so I'm going to pause there on talking about Pdbkb. I just put a slide in here just so that people are aware of where to go for other training resources related to PDB and Pdbkb. So we have a pdb.org/training page where we have lots of tutorials available. And we also have a YouTube channel where we make lots of webinars and other videos available as well. So just to give you information that there is more available, and if you want more information, you can contact us. And the other thing, just before I go to the tutorials, is just to mention that we have a position actually available for a bioinformatician or scientific developer to work at PDBe alongside Kemble and CCDC on the Biochem Graph project, which is what Preeti will be talking more about in the next session. So this closes in a couple of weeks' time on the 3rd of May. So you can apply at pdbe.org/jobs. But if you're interested, then pay attention to what Preeti talks about and see if it sounds interesting to you. So I wanted to go through the Pdbkb tutorials and where to find these. I don't know, Chris, are there any other questions that it's worth me addressing before that?
You're all up to date.
Okay, great. Okay, so as I mentioned, we have some tutorials for Pdbkb to help you understand how they work, how to get information from these. I've added the link here in the slides, so hopefully, because it's a nice short link, you can quite easily get to that. So bit.ly/RSC_PDBekb, and this should take you to a tutorials folder where we have a couple of different tutorials. So we have a basic tutorial on Pdbkb protein pages, how to access data and look at interactions. We've also got some nice advanced tutorials that Preeti put together, which are looking at specific case studies. So looking at either ligand interaction data, macromolecular interaction data, how to find additional functional information. And in the tutorials folder, they should both also have answer documents, so you can run through those tutorials and check against the answers. So what I will do is just click on that link and show you where these are. So these are what you should see by going to that tutorials link. I hope, Chris, are you able to access that okay?
Yeah, no problem.
Great. So the first ones I mentioned, we have these Pdbkb worksheets. So these are quite nice and visual. They take you through how you access the pages, the type of data you find, and also a bit of information about the visualization tools we have on these pages and what they mean. Then for this particular example, which is myself, this P53041, which is a phosphatase protein. We'll go through the interaction section to see where the coverage for a particular individual entry is. And then you can look at answering the questions about the different domains that are available. There's also a section here about the ligands section as well. So finding ligands and understanding what we mean by cofactors and what interaction annotations we add to ligands, and looking at the interaction patterns between these ligands. So one of the things that I didn't show you in the slide is actually you can show the ligands section collapsed. If I just zoom in slightly here, you can collapse the ligands section, and what you can actually see by doing that is quite clearly see where these common interactions for different ligands, highlighting where they're binding in the same position. And so some of the questions here ask about, why is this the case? And hopefully the answers can help you through that as well. We also introduced the 3D view as well, to open the 3D viewer and show you how you can actually show individual visualization for a particular protein structure. So if I then just show you the more advanced worksheet. So this is split into 3 sections. So the first section is on ligand binding sites, and these each use a specific Pdbkb page as an example. So I can open that and show you where, if I can move my sharing box. So this shows you that particular, goes to this particular Pdbkb page, and all the questions are then related from this particular page. It gives you a bit of a background of the case study in this example, and then it runs you through tasks where what we're trying to do here is, rather than just say this is what you do, it's kind of getting you to think about how you navigate these sections with a few hints to help you work out how. And hopefully it's a bit more, although it's a bit more advanced, it might help understand the problems a bit more, understand how you can interpret the data from Pdbkb pages. So we touch on similar scaffolds between ligands here. We also look at the functional annotations, and we integrate other resources such as CANSAR, which actually provides additional interactions from predicted interactions for drug discovery. And we also look a bit at the superimposed binding as well, and how you can use those to go alongside your information from the interaction section. So yeah, I won't go too much through the other sections. I think obviously for this, for chemistry related things, the ligand binding sites I think is probably the most interesting section, but we also have one on the macromolecular interactions parts as well, and also another one here which is based on some of the additional functional annotation on cofactors, active sites and things like that, how you can get more information from that, and using in this case the example of COX-2 and the inflammatory response, and obviously that is a key drug target for preventing the inflammatory response. So yes, I think I will leave it there, and let me stop sharing my screen. I guess ask if there are any questions at the moment.
Yes, there's an interesting one for you here. How would one supply annotation data to Pdbkb? Are there formats and processes? Also, if you find any information to improve the data, how is it best to supply that?
Okay, so yes, there is a deposition system for Pdbkb. If you go to Pdbkb's homepage, so that's at pdbkb.org, and Preeti's actually put the direct link in the. Oh no, I think I went to panelists. So yeah, Preeti will put the link on the chat for everyone to access. There's information of what is required for those particular links. I could probably, maybe I'll share my screen just to show you what that page looks like. Yes, so there's a page here for guidelines for collaborating. What that term means, how you provide annotations. Generally, there are some rules behind this because it's a sort of semi-automated deposition process. So generally this is designed for having a collection of annotations that you then want to relate to the Pdbkb pages. So most of these providers are people who have developed their own resources or have their own large data sets of data. But again, if there's anything else that is missing or that you think you're not sure if you quite qualify for this, then it's worth contacting us at PDBe and we can discuss how we can incorporate more data as well. Did you add anything else on that at all, Preeti? Okay. Any other questions to answer, Chris?
No, you're all up to date at the moment.
Excellent. Okay, so we're actually slightly ahead of time. So we can have a slightly extended break. We were due to come back at 2 to 3. But I guess I'll hand over to you, Chris, to introduce the sponsors.
Bailey, do you want to say a few words?
Yeah, perfect. Can you all hear me okay?
Yeah, sure.
Perfect. All right. First of all, I'd like to say thanks to David so far for the excellent and informative workshop that we've had. And I hope that Preeti can add some more value to that as well as we go into this second session after the break. As you may be able to see from your screen, my name is Bailey and I work for Liverpool Chirochem. We're a chemical technology innovator on a mission to accelerate small molecule drug discovery through expanding access to 3D chemical space. And we're proud to sponsor these events and these series of workshops because we understand how essential these open source tools are for the global research community. At LCC, we specialize in using stereo-defined N heterocycles for use in DEL, fragment-based lead discovery, linkers for heterobifunctionals, and our 3D discovery virtual library. If you would like to find out more about how LCC could support any of your projects, then please reach out to us through the contacts page of our website, which is liverpoolchirochem.com, or you can find us on LinkedIn. I hope you all enjoy the rest of the workshop, and thank you very much.
Okay, thanks very much, Bailey.
Okay, I'll just start sharing my screen, and hopefully you are able to see my screen.
Yeah, all looks fine.
Perfect. Okay, so hello everyone. So today I'll be talking you through our new project, relatively new, called as Biochem Graph. It is essentially focused on small molecules and sort of, integrating the knowledge graph to enhance the information of small molecules in PDB. And to give you a brief overview of today's presentation, we'll start with the first half with the project introduction. Then we'll move on to what existing information we have for small molecule in PDB. And towards the end, our efforts through this project, how we are enhancing the current information on the small molecules. So to give you a bit of flavor or the motivation behind this project, back in 2019, we carried out an extensive public survey, and we asked a variety of users what are the frequent scientific questions they usually ask related to small molecules, and where do they get their data from, and what kind of annotations they are generally interested in. And through this survey, we got almost around over 120 responses from industry and various academia representatives. And this survey told us, indeed, there was a need for a common resource which could integrate the information. So this project came into action. And this is essentially integration of data between ourselves, PDBe, Chemble, which is again a database for manually curated, having bioactivity data for drug-like compounds, and then CCDC, which is Cambridge Crystallographic Data Center, which is experts in structural chemistry data. So with this project, we would like to map the data resources from various for a given ligand and improve the annotations. And in the second half of the project, what we would like to do is, expose this data in a more meaningful way. And we have talked, you have listened to David talk how Pdbkb efforts has been in place to give more structural context to the data. And the same we would like to do for ligands so that users can see the annotations more clearly in the archive. So this is just an example to show you when I say, integration of the data, what I really mean. Here I have taken an example of a painkiller codeine, which binds to various targets. And you can see in Chemble, and Chemble has, in fact, a lot of target data for this codeine. For instance, it binds to human kappa opioid receptor. In PDB, we don't have the 3D structure for codeine, but we have similar ligand, which has same scaffold, two of such molecules. One is present in Chemble, and the other one is not. Now, both of these structures they are bound to the same target, which is kappa opioid receptor. And if you look at the binding site where these two ligands are bound, if you superimpose the binding site, you would find that they are essentially the same residues which are bound in both the ligands. And this kind of gives an idea that codeine might be also binding in the same pocket and probably involving same residues. And then, CCDC has the 3D structure for codeine, and their information about, the binding affinities and functional groups can further enhance the information about codeine. So at the moment, you can do this manually. With this project, we would like to establish an automated pipeline which can do this, and such transfer of annotations between various databases can further help in target validation and drug repurposing. So to give you a brief summary of Biochem graph, basically this is a BBSRC, BBR funded project. It is a 3-year project which started back in 2020 end, and it is going to go until 2023. It is a collaboration between PDBe, CCDC, and Chemble. From our side, PI is Samir Valenkar, and co-PIs are Andrew Leach from Chemble and Ileana Grian-Greco from CCDC. Now, one of the high-level project goals of this project is essentially to improve the data representation of small molecules. This essentially includes, data standardization. For example, the way we represent data in our MMSIF files, to expand on those, accommodate annotations like scaffolds and other functional annotations of the ligand. Secondly, we would also like to improve the integration of the data between all these 3 resources. And third main goal is to expose this data on novel web pages, and also to develop visualization tools, which will help users to better visualize small molecules. The deliverables of this project includes 6 main things. First, we would like to increase the overlap between PDB, CST, and Chemble small molecules. Next, we would like to identify and integrate the common target information and its associated bioactivity data. We would like to characterize the binding site of the target protein based on ligand interaction status and the structural and sequence similarity. Moving on, we would like to enrich PDB data with new annotations, further distribute all these expanded annotations and data in form of probably a knowledge graph, and then display this ligand data on aggregated views. In this talk, I would talk more about the deliverable 1 and deliverable 6 to you. So before going into the details, let me just give you a quick overview of what we have currently in the archive, in PDB archive, in terms of small molecules. So the first thing I would like to highlight are the CCD, which is essentially the chemical component dictionary, and it defines the unique chemical compound in PDB, and usually they are 3-letter codes: ATP, MAN. For ATP, it's ATP, MAN, heme, and they are essentially represented as MMSIF file. To date, we have over 36,000 chemical compounds. Then we have specific use cases like BIRD or PRD, which is essentially biologically interesting molecule reference dictionary. These are specific to peptide-like antibodies and inhibitor molecules present in the PDB archive. And they include almost over 1,000 representatives. Very recently, we also did carbohydrate remediation, where we basically defined the carbohydrates in the archive as a whole molecule rather than their individual components. And some of these carbohydrates, they are also BIRD, for example, sucrose. While the scope of BIRD and carbohydrate polymers is limited, CCD sort of overall covers the entire archive, PDB archive. However, it has limitations. For example, in this particular PDB entry, I show you 6LQ4. It's essentially a structure for acyl-CoA dehydrogenases, FADE5. Now, here it shows the structure has been bound to 2 ligands, MYR and CoA. However, if you go and look into the structure in the 2D or 3D viewer, you will see that this is in fact one single ligand. It isn't 2, and it is covalently bound. If you go into the literature, this is essentially myristoyl coenzyme A, which is a substrate for this particular enzyme. And just because of the way we represent the current data, it has been split into 2. Now, we would like to correct such cases and try to address this issue. And via this project, we have been trying to do so. So what we did was we identified something called as bound molecules. This is essentially all the molecules which are covalently bound in a given across all the PDB archive. And this would essentially represent chemically complete ligands. And we use RDKit to get a list of these bound compounds, and they can be described by unique INCHI and INCHI key, all the covalently bonded chemical compounds. And since it's an automated process, it is much more maintainable, and it expands over, sort of complements the information which we have for BIRD and carbohydrate polymers at the moment. In our analysis, we found over 5,000 unique bound molecules. So for example, now you can see from our process, which identified bound molecules, this compound, myristoyl-CoA, was now detected as one single compound. And here you can see it's a 2D image and the INCHI key. In fact, now because of this, it was for the first time that we were able to map myristoyl-CoA to other databases For example, in Kiwi, we can link now to Campbell data, drug bank, PubChem data, and various other external databases, which was previously split into two. So this gives us an advantage of increasing the overlap of the current molecules in the archive, and also enriching their annotations. So to give you the numbers, to what extent it increased the mappings, out of approximately 5,000 new entry keys or bound molecules, we got almost 172 exact matches to the Campbell data. And there were many other matches which was matched only by the first part of entry key, which corresponds to the connectivity. And we did a detailed analysis of these cases and found that there were some genuine discrepancies because of differences in isomers, and sometimes it can be a wrong or missing stereoisomer either in Campbell or PDBe. So this pipeline also gives us a medium to correct the annotations, misannotations, and give a uniformity to the data. On this table, you can find the other increased mapping. There were a lot of cases which was increased for KEG, drug bank as well, Brenda as well. So now, let's moving on to the advantages of doing this analysis or defining these bound molecules. First of all, we can recognize all the covalently bound compounds and treat them as single compound. This will help us in finding them and representing them. It can help us identify, if a ligand is a part of a bigger bound compound. And of course, it will increase the mappings to external databases. Apart from identifying this, we can also standardize the data to represent these molecules. And also, this also gives us a fair comparison to compare the bound molecules in a more biologically relevant environment. Apart from these bound molecules, we also enhanced our information for scaffolds. So we identify Markov scaffolds and fragments for each chemical component in the archive. This is done weekly, and as a part of a weekly process. And here I have shown you the example of cobalamin, showing the Markov scaffold and all its various fragments. All this data is available on our FTP and is updated weekly. And this particular information is also represented in RDKit standard representation in zip file. And here I have given a link for you in case you want to access this information. And so all this information which we do is basically by an in-house code, which we call Pdbe CCD utils. This is essentially a wrapper around the RDKit, and this is open source code. And any one of you can go to the GitHub, download the code, and use it to do the exact same things, identifying scaffolds, fragments, even similar ligands to a given library, and create your own images. We use it every week to update our data and calculate 2D images, idealize coordinates, various physiochemical properties, as we have linked it to Unichem pipeline, which essentially maps the chemical components between various databases. So this is how the in-heart core powerhouse of our chemical components. And then we also have various functional annotations of ligand in our database. Every week we map, each chemical component dictionary (CCD) to various databases. For example, to get an annotation for small molecules which can act as substrate or a product, we go and map to RIA or KB database. RIA is essentially a database which stores the information for all the biochemical reactions with each participant's, what are the exact components of the reactions. So from there, we fetch the information if a particular small molecule is behaving as a substrate or product. We map drug-like ligands from drug bank database. We have a pipeline called Cofactor Pipeline, which again annotates the ligands as cofactor. All this data is now available via our API and also on the protein pages. You can see here I've shown you the images, and they are highlighted in the ligand gallery for a given protein page, highlighted in green, and you can spot the cofactor or drug-like molecules very easily. Now we would like to transfer this particular information and create new views called as Pdbkb aggregated view of ligands. As you have heard from David's talk all about Pdbkb efforts in this project, one of the major aim is to create these aggregated view of ligands, or we call ligand pages in short. And one of the major goal is, of course, to aggregate the data for a given ligand across all the entire PDB archive to make the ligand data fair, which is to make it more findable, accessible, interoperable, and reusable, present it in a user-friendly way. And these pages will eventually replace the current service which we have for ligands, Pdbkb. And all these pages will be the integral part of the PDBe web portal and will be, sitting in the umbrella of Pdbkb. Now, to give you an idea about, I have already talked in the beginning, like how we have focused the entire project on the extensive user survey. For these pages views as well, we have, created various user personas, and for each users, we have jotted down what kind of data they would like to know and what they are interested in. And then based on the kind of data each user type is interested in, we have tried to create a list of scientific questions which we would like to answer from these aggregated view of proteins. And here, all in this yellow, you see are the things which we are planning to answer via these set of pages. The ones shown in greens are nice to have, but not our priority at the moment. And however, one of the major challenges while designing these aggregated view of proteins, while compared to protein pages, is the enormous amount of data. For example, there can be a case where there can be more than 18,000 similar ligands, which would essentially mean would you like to see all those 18,000 similar ligands? How would you expose such a data? How will it make sense to to this data? Another thing with these ligand data is it has a different way of looking at it. For example, if you talk about interactions, you can look at interactions from a ligand perspective as well, and from protein perspective as well. And one of the challenges is, how we would display all this information in a meaningful way, and it would be easier to understand. And this is why we have sort of today's next session, which is, the ligand pages design session or user testing session. In this session, we would be sharing a bit of more, mock-ups and an evaluation form, and we can talk about it a bit more into the details. But first, I would like to stop here and ask if there are any questions, and then we can go into the details about design session a bit later.
Okay. Can I ask a quick question? You talked a lot about web pages and things like this, but is there going to be programmatic access as well?
Yes, so most of the data which I've talked about is already there, accessible, because it is there in the database. It is accessible via our API endpoints, and in the graph database as well, it is available. And web pages is, making it accessible via creating a novel page and creating new visualization tools to basically improve visualization and better understanding of the data. But most of the data which I talked about, like, scaffolds, even similar ligand data is there in the database and can be accessed via API.
Okay. But at the moment, the sort of Kemble and PDB are independent, aren't they? So you can't access all the information from one point.
Yes.
Is that correct?
Yes. Yes, that is correct. And that is one of the goal of these pages as well. So for example, you will see in one of the prototypes later in the section, we have tried to cross-link a given molecule to other databases as well, and sort of pick up the annotations as well. For example, highlighting straight away that, hey, this ligand is a drug, hey, this ligand is, a reactant, and giving it more functional annotations.
Yeah. I noticed most of your linking of structures was via Inchy keys.
Yes.
Yeah. I mean, do they lack stereochemical information? So you can get problem with cross-linking or cross-linking
yes.
isomers
yes.
and things like this.
Yes. And that's what you saw when we were mapping the bound molecules to the exact matches in Campbell. Majority of them had the issue with the stereoisomer. But we are using standard Inchy keys to generate the bound molecules from the RD kit.
Yeah. And another question occurs to me. With covalent ligands, what you have in the PDB is the product. You don't necessarily know what the ligand was before it reacted with the protein. So
yes.
what do you cross-reference to, the original ligand or what?
I guess maybe I'm being on the curation side, I'm probably slightly better placed to answer this. So at the moment, generally, what we assign a ligand to with curation is what is in the structure itself. So that's generally the product, what we actually observe in the particular structure. We have actually discussed a bit recently about how we can capture information about the precursors to the reaction, and also find a way to link that data together as well. But at the moment, we don't have that information. And we do also have some inconsistency in the PDB archive about which one is defined, whether it's generally it's the product, but in cases where perhaps the initial reactant is only losing one atom, then in some cases we actually define the original reactant with a leaving atom defined. So it's not ideal at the moment, but it's something we're looking into.
Yeah, I can see instances like where you have a electron deficient aldehyde or something like this, where you get a hemiacetal formed with a serine. What do you define as the ligand? Is it the aldehyde or the hemiacetal? Yeah,
exactly. And generally that's why we use the product, because it's the best way to validate the geometry of the model by defining what they actually have modeled. But then we are losing the information about the original drug molecule perhaps that was added, and that's the issue.
Yeah. Okay. There's no questions in the Q&A, so maybe carry on.
Okay. Okay. So let's open so here I have a prototype for the current mockups which we have. Let me just open it for you guys, and I will also put it in the chat box so that it is easier for it takes a bit for loading, but
yeah, okay, it seems to have loaded now.
Yeah, but once it's loaded, it should work pretty smoothly. It is not a static web page. We have made this using an online tool called Figma. So if you can actually pretty much scroll through the images and interact with the page. Not everything is clickable, but if you would like to know what is clickable, if you click around any white space, the things which are clickable would be highlighted in blue. So then you can know, okay, this is clickable. And in this current prototype, we have the 4 sections: description section, structure section, interaction section, and related ligand sections. Tab or page, you can say, which are working, and you should be able to interact with them. And we have tried to, on the slide, you have an evaluation form, which is a Google form, which basically is for us to collect the data, how these mockups are. So the way we do these user testing is essentially we ask a given, like, hey, are you able to find, is this particular compound is drug or not? So it's more like yes and no questions, and all you gotta do is walk through the site. And here, this mockup is for STI, the drug STI, and then you can see the description. You can pretty much scroll and see what we have data for each of these sections, and then try to answer the questions which we have in this form. In the first section is just for us to know what is your background, so that we know the people who filled in follow in which user category. But then if you go here, you can see, like, for every tab, we have a questionnaire. For example, for description section, can you find the mapping between the ligand and other databases? And then you have this MCQ, which you can, fill, and then let us know how the entire exercise or how the page interaction was with you, was for you. Was the data more, understandable and easy to navigate? Just to emphasize over here, this is not a test of a person, but it is a test of a prototype, and it is the test for us. And there are no incorrect answers. And the whole thing, if you just interact with this form, this is just going to take like 15 to 20 minutes. But I would leave you to it, and if you can play with this mockup and let us know what are your thoughts, and we can discuss towards the session as well, how you found the pages. If you have any suggestions or feedback for us, that would be great. And yeah, so I guess I can leave you to play with this prototype for, let's say, 10-15 minutes, and then we can come back, we can sort of discuss.
Do you want to put the link in the chat or something?
Yes. Yeah. Please.
It's got it, yes.
And then the final one.
You got it.
So we have put both the link to the prototype as well as the feedback form. And yeah, have a go at it. Yeah.
I was just going to add, just if anyone has any other questions about anything we covered today, then please add them to the Q&A chat box, and we can discuss those.
Mm-hmm. It looks like the click to view in 3D button's not working. Is that correct?
Yes, that's correct.
Yeah.
So you can see the links which are working. If you
Yeah.
click on it, let me just show you. The blue thing will highlight. So not all the buttons are workable, but
Yeah.
to give you an idea how it will work. And this is how we envision, currently the pages. But this is why we want to have a bit of user testing as well, so that we know if what we have made so far makes sense. So if you can, so if you click anywhere over here on the screen, you can see which things are clickable in blue. So for example, over here, you can interact with page.
Yeah, one thing I would request I would have is a button to copy either the smile string or a mol file to the clipboard.
Okay, yeah, that's actually a nice idea because they can be very long, and I can
They can be,
imagine.
and you end up picking up sort of bits of non-printing white space and things like this.
Yeah.
There's a question in the Q&A. Is there a tool you recommend for creating a ligand in PDB format?
I guess I can answer that. It's tricky to recommend necessarily because it depends why you're creating the ligand, I guess. So there are various tools in the case of structure refinement for creating ligands that are then used to refine into a structure. So that can be done using, for example, programs like Coot, or also within the refinement softwares themselves. They actually use PDB ligands as definitions that can then be dropped into structure for refinement. Obviously, there's also the option to make ligands separately if you just want to use them for docking or things like that. I don't know, because it's not something that I've ever done. I don't know any necessarily the best tool. I know it can be done using viewers like Coot and Pymol. I'm not sure whether there's maybe newer specialist tools for doing this. The alternative as well is tools like ChemDraw, where you can create a ligand using the 2D diagram and then export that information as smile string and ligands and things like that as well. So there's lots of options to this question. So I'm not sure about necessarily the best way to do it. But yeah, I think the first thing is to kind of define why you want to create the ligand and what you're using it for. And then I think the tools can be more specific for that.
Yeah. I mean, I think one of the tool which is, probably the best for creating different file formats is OpenBabel, which is a command line tool, but it will convert any file for almost every chemistry file format into any other file format. So it's a way of creating, if you have a smile string, you can create a PDB file or whatever. Most drawing packages would probably save the 3D structures in SDF format, but you can then convert that into a PDB format.
Yeah, there's like I say, there's almost such a wealth of options that not having used enough of them myself, I don't want to say which one's necessarily the best option, but they say it's the ease of doing it. In some cases, you might have to then convert file formats separately and that side of things. Whereas, yeah, OpenBabel is something we use kind of in the background for PDB data to generate descriptors and things like that. So, yeah, it's a very kind of all-encompassing software, but command line based rather than browser.
Okay. I presume you're happy with people to use that form to submit their responses after the event as well?
Yeah. Yes. Yes.
Yes, don't feel like you have to rush to do it now. It would be nice if you want to take some more time, then please do. I think this is just a chance to show you what these pages look like, and maybe if there are specific questions that people want to ask during this session, then it's a good opportunity for us to answer them. But please take your time to look through these outside of this meeting.
Yeah. And if something comes in your mind, like, hey, I would like this data to be here as well, you can let us know as well. Like if something you think is missing in these pages and you would like to add or see, you are more than welcome to, tell us now or later in the forms as well.
Yeah. So I've just tried to look at the form again, and now I get you need permission. This form can only be viewed by users in the owner's organization.
Is it? Oh, that's weird. Let me see. Let me check.
Yes, that's someone else has also mentioned that. Okay.
Sorry for that. Let me quickly fix that.
Do it. Let's try this. I think I've got the
Is it faster? I think you also have the permissions.
well, let's try this link. Let me know if this one works, but whether it has the same issue.
No, it says you need permission.
Okay, it's still the same problem.
Yeah. So one of the things that you said was sort of out of scope was the CANSAR and things like this. I always thought they were really very, very useful because they were very much therapeutic focused.
So was this to Preeti rather than Bob?
Well, I'm not sure who I think it was.
I can't remember who said what.
It was on one of the slide. It was one of the green things, I think, on one of the slides. So it's not part of it, but I mean, I know it hasn't been updated for quite a long while, but it was quite an interesting way of looking at the data. I guess that's more of a Kemble thing, wasn't it? But
we do definitely integrate CANSAR data into our protein pages. So that is we do integrate CANSAR data. I'm not sure the specifics of Biochem Graph how we integrate CANSAR.
At the moment, only at Pdbkb pages. Yeah. So if you go in the functional annotation section, you can see the cancer data on the ProtVista viewer.
So yeah, I still get you need permission, I'm afraid.
Okay, even after the modified, it's not good because I just updated the permissions.
Sorry, we're just troubleshooting at the moment, see if we can work out. We shared it with each other. We should have shared it with someone outside of EBI.
Yeah.
Yes. It's one of the perils of doing things live, isn't it?
Yes, always.
We can always share it afterwards anyway. We'll be sending out a link to all the people with the video link and things like this. So once it's sorted, we can include that as well.
Okay, perhaps that's maybe we'll work on this after the session, and
hopefully,
and then we can send out the link with the, like you say, when you email the attendees afterwards.
Yeah, sorry for the inconvenience.
Yes, don't worry.
Is there any specific question which anyone would like to highlight while going through these web pages or prototypes? Maybe we can quickly go and discuss the interaction section, and then let me share my screen, and then probably we can use that as a starter for conversation. And there is something, we would like to also get your perspective into the interaction section a bit, because like I mentioned before, interactions is something which has different meaning in different contexts, and it is a lot of data as well, and we would like to represent it in more user-friendly way and more meaningful way. So in this section at the moment, we have taken all the instances of a given ligand from the entire archive, and we have sort of calculated the interaction frequency for a given ligand, considering all the instances of a given protein. And interactions, the way we describe interactions is essentially by a tool called Rpgo. This one was developed from Tom Blundell's lab. And they have different type of interactions as well. Like, they define hydrogen bond, covalent bond, which at the moment we don't show in this page, but in future we will. But at the moment, if any kind of interaction, we treat it as just one interaction. And this is just to show or give people an idea about, what are the consistent interaction of a given ligand across when it's in general. So, and here you would see for a given ligand, you would see a 2D image with contoured lines showing you the strength of contour lines will show you the intensity of interaction frequency. More the number of interactions, more dense or the contour lines will be. So here you can see, like, for example, this particular is the hot spot for this particular ligand. And here you also see it for each ligand atom, you see the frequency of that particular atom, interaction frequency. And then you can see for various amino acid type, what is the interaction frequency. And people can also sort the data based on interaction frequency strength or your amino acid property. So for example, maybe perhaps collating the amino acid property, it can somehow give an idea if it is binding only to positively charged amino acids or hydrophobic amino acid and whatnot. And in reality, the heat map on the plot vista viewer, which you see over here, would be communicating with your 3D, the 2D image as well. So for example, if I click over here, C12 atom, you can see the C12 is highlighted in the image as well. And in this mockup, you only see over one atom, but essentially in this heat map, on any box you click, it should highlight the atom. And this is just to show where spatially that particular atom is located. Users can further filter the data, like, okay, I only want positively charged residues and stuff like that from this section. So this is what our perspective is for this interaction section. My question to the users is that something which is apparent, like I have from when you see it, is it apparent enough, or do you think we can improve on this view? Or do you have?
I think if you can show the interaction types as well, whether it's a hydrogen bond or hydrophobic interaction, that would be useful. Or
so this is just the first prototype, but in the long run, we plan to put a filter criteria where people can filter based on interaction type as well, main chain interaction, side chain interaction, or hydrophobic interaction, and the way you will choose the interaction type, the heat map will change accordingly and will only show you just those type of interactions. So yes.
I guess the other thing is that many crystal structures have important water molecules as well. Would you include that as well?
Yes. So in fact, in this mockup, I only show amino acids, but the plan is to show also the nucleic acids as well. So include DNA and RNA, and water molecules are difficult to handle at the moment, but we are thinking about that as well to characterize water-mediated interactions as well.
Yeah, I think one of the things that there's one of the great things is we have so much data, but the downside is not letting everything get too crowded and make it kind of difficult for people to be able to use the visualization tools. So yeah, we want to add more of these things, and it's working out how to add the visualization without complicating what's already there as well. So I think what you said a good point. We have information, like we say, on bound molecules and interactions with other, and for example, on our ligand environment pages, we do show those interactions with the water molecules from the ligands. But it's presenting it in this way with all of those different types of data is a challenge we're still kind of working how to do.
I think this looks really interesting. Would you envisage you will be able to query on this? Have a, I'm looking for mimics of an amino acid, or you've got here a piperazine interacting with a glutamate or something like that.
Sorry, could you ask? Yeah, I'm not quite sure what you mean. Say that again. Sorry, Chris.
So, I mean, this is a way of viewing it, but would you be able to also query? So, say, draw a structure and say, I would like to see examples of this structural motif interacting with these types of amino acids.
I think maybe that's a bit further down the line. One of the things is obviously we're trying to highlight scaffolds and similar ligands with the same scaffold. So I think initially it would give you the option to see all the data for all those related ligands, go to the ligand page for each one. I think you're right. It might be quite nice if we can find a way to then aggregate the data for scaffolds. Maybe is almost the next point, but we're not quite there yet, unfortunately. Although the data is there in the background, as Preeti was saying. So in theory, it's programmatically possible. It's not something we've quite got our heads around in terms of doing it for the full archive and visualizing it as well, which is going to be a challenge.
Yeah, but users can still go, and even if these pages are not live, but if you are interested in, getting similar ligands to a given CCD in PDB archive, you can go to our API, and you can see, so for example, in the related section, you see similar ligands, and you see, similar ligands with more than 60% score. And this list you can still get from API. It won't be in forms of pictures as beautifully presented here, but people can still go and access this information. And the way we currently measure similarity is via a parity method. And based on that, we define anything which is greater than 60% we show on this page. And using our PDBe CCD utils, users can go and identify scaffolds as well for using a given library, set of libraries of chemical compounds. They can also, do Markov scaffolds as well.
Yeah. Okay. Well, there's no open questions, so I think you've answered everything as you've gone along, which is really nice.
I found the setting. I found the setting, and I think, it was restricting responses to EBI, and I hope now that's not the case. So could you just give it a quick try and let me know if that's? I'll try myself on that.
Yeah, I can see it now.
It was more hidden than I would have expected it to be, but there we go, found it. Okay. So obviously take the time to respond to the form and have a look through and give us your feedback.
Yeah. Okay. As I say, there's no open questions, so it just remains for me to thank you for giving these presentations. I think the whole PDB is absolutely a fabulous resource. I use it every day, and I'm just grateful that it's open and freely accessible. I don't know what I would do without it, to be honest. So as I say, thank you for everything. It's been a fantastic presentation again. I'll be editing the video over the next few days, and hopefully it'll be on the YouTube channel early next week sometime. Okay.
Excellent. Thank you very much, Chris. Thanks everyone for attending.
Thank you, everyone.
Right.
Bye-bye.
Bye then.
Thank you very much,
Bye.
guys.
Bye.
Bye.